Hello students, I am Professor Rashmi Pingle from Zoology Department, KBN Naik College, Nasik. Students, today we are going to discuss about the Kreb cycle. So before going to directly to the Kreb cycle, first of all we will see what is the respiration. Student, respiration, especially the cellular respiration, is the procedure by which oxygen is used to oxidize the organic fuel molecules like the lipids, protein and carbohydrate, and it generates energy in the form of ATP, water and carbon dioxide. So the respiration is basically of the two kinds, aerobic respiration and anaerobic respiration. So aerobic respiration means the respiration which takes place in the presence of oxygen, as in eukaryotic cell and some prokaryotic bacteria. And anaerobic respiration takes place in the absence of oxygen. So today Kreb cycle is a part of aerobic respiration, is a second step of the aerobic respiration. So we will move to the aerobic respiration. As I told you, aerobic respiration is a respiration, means is the breakdown of food in the presence of oxygen. So the fuel molecules for the oxidation, they may be the fatty acids. Fatty acids are formed from the breakdown of the lipids. They may be the amino acid formed from the breakdown of the proteins, and it may be the glucose. Chiefly the glucose is the major source of energy in our cell. So when the fatty acid and amino acid are used as a source of energy, in the first step of the aerobic respiration process, they are firstly converted into the two molecules of the acetyl. They are converted into the acetyl-CoA. They are converted into the acetyl-CoA. Fatty acid are converted into the acetyl-CoA and amino acid. In case of amino acid, as you know, amino acid may be ketogenic and they may be glucogenic. So glucogenic amino acid, they may be converted to the pyruvate, and the ketogenic amino acid, they may be converted to the acetyl-CoA. And in case of glucose, which is used as a chief source of energy in our cell, is converted firstly in the pyruvate. The conversion of glucose to pyruvate takes place through the pathway known as the glycolysis. Glycolysis is the first step in the process of the aerobic respiration, and glycolysis takes place in the cytosol part of the cell. So, after the formation of the pyruvate before entering to the Kreb cycle must be converted to the acetyl-CoA. So, pyruvate is converted to the acetyl-CoA through the PDH complex. PDH means pyruvate dehydrogenase complex. Here it is called as complex because many copies of enzymes are present and 5 coenzymes are involved in this, therefore it is known as a complex. So the two pyruvate molecules are converted into the two molecules of acetyl-CoA, and this reaction takes place in the matrix of the mitochondria, and it is also known as the linking reaction. Linking reaction means it links the glycolysis cycle with the Kreb cycle. Hence it is known as the linking reaction. Then after there is formation of the two molecules of acetyl-CoA, this acetyl-CoA are fed into the Kreb cycle. They enter the Kreb cycle, and there is complete oxidation of this acetyl-CoA to carbon dioxide, water, ATP, and also reduced coenzymes such as NADH and FADH2 are formed in the process of the Kreb cycle. And this is the second step of the aerobic respiration. And the Kreb cycle, location of the Kreb cycle is the matrix of the mitochondria. And again, the reduced coenzyme NADH and FADH2, they need to be used again. So they need to be converted to their oxidized form. That is, NADH should be converted to NAD plus and FADH2 should be converted to the FAD. And this oxidation of the reduced coenzyme takes place through the third process of the aerobic respiration, that is known as the electron transport chain. And this ETC takes place in the inner membrane of the mitochondria. And through this ETC, lots of energy in the form of ATP is generated. Now student, after knowing the process of aerobic respiration and after knowing the steps of the aerobic respiration, as I told you, there are 3 steps of aerobic respiration: glycolysis, then the linking reaction, second step is Kreb cycle, and lastly ETC, that is electron transport chain. Now we will move toward the Kreb cycle. So the Kreb cycle was the first cyclic pathway to be invented. It was the first cyclic pathway to be invented, and this pathway was invented by the Sir Hans Kreb. The scientist name Sir Hans Kreb. Hence it is known as the Kreb cycle. And it is also known as the citric acid cycle because the first intermediate formed in the Kreb cycle is the citrate. Therefore, it is also known as the citric acid cycle. And it is also known as the TCA, that is tricarboxylic acid cycle, because the first intermediate citrate of the Kreb cycle has the 3-carboxylic group in its structure. Tri means 3 and carboxylics means COOH group. So if we see the structure of the citrate has the 3-carboxyl group present in its structure. Therefore, it is also known as the tricarboxylic acid cycle. Now, the location, as I told you, it takes place in the mitochondrial matrix. So now when we see the structure of the mitochondria is a double membrane structure. The outer membrane and the inner membrane is convoluted, is folded to form the cristae, and inside that inner membrane the matrix of the mitochondria is present, and in the matrix of mitochondria the enzymes for the Krebs cycle are present there. So Krebs cycle takes place in the matrix of the mitochondria. So now let's move toward the steps of the Krebs cycle. Student, there are total 8 steps in the Krebs cycle. So in the first step, the oxaloacetate is a 4-carbon compound. Oxaloacetate is a 4-carbon compound. It is present in the matrix of the mitochondria. And when the acetyl-CoA donates its acetyl group to the oxaloacetate, this acetyl-CoA come from here, the linking reaction. So the acetyl-CoA donates its acetyl group CH3C double bond O group to the oxaloacetate, and the condensation reaction takes place and the coenzyme A is released and the citrate is produced. Citrate is a 6-carbon compound and the enzyme responsible for this conversion is citrate synthase. So there is the formation of the citrate. This is the first step of the Krebs cycle. Now, in the second step is converted into 2A and 2B. So what happened in the second step? Citrate is converted into the isocitrate. Citrate and isocitrate are the isomers of each other. Isomers means they are having the same molecular formula but the different structural formulae. So the enzyme responsible for this conversion is aconitase. And aconitase did this conversion in 2 steps. Citrate, at first dehydration of citrate takes place. Dehydration means the removal of water takes place and cis-aconitate, an intermediate is formed. Then there is hydration of the cis-aconitate. Student, hydration means the addition of water, and again enzyme responsible is aconitase and the isocitrate is formed. Isocitrate is again a 6-carbon compound. Then in the third step, isocitrate is converted into the alpha ketoglutarate and the enzyme responsible is isocitrate dehydrogenase. Dehydrogenase means it is responsible for the removal of the protons, for the removal of the hydrogen ions, means for the oxidation. Removal of the hydrogen ions is known as the oxidation. So in this process, oxidative decarboxylation takes place. Oxidation means removal of the protons and decarboxylation means the removal of the carbon dioxide. So here first carbon dioxide molecule of the Krebs cycle is released and first NADH is generated, and here the hydrogen ion acceptor is the NAD plus, and then isocitrate is converted into alpha ketoglutarate, which is a 5-carbon compound, as one carbon is released in the form of carbon dioxide. Now in the fourth step, what happened? Alpha ketoglutarate is converted into the succinyl CoA, and the enzyme complex responsible for this is alpha ketoglutarate dehydrogenase complex. Here also oxidative decarboxylation takes place. Oxidation means removal of the protons, removal of the hydrogen ions takes place, and here also NAD plus is the hydrogen ion acceptor, and decarboxylation means removal of the carbon dioxide takes place and coenzyme A is added. So therefore alpha ketoglutarate is converted to the succinyl CoA, which is a 4-carbon compound. Now in the fifth step, what happens? Succinyl CoA is now a 4-carbon compound because one carbon from alpha ketoglutarate has released in the form of carbon dioxide. So here the second molecule of carbon dioxide of the Krebs cycle has released and our second NADH molecule has formed. Now succinyl CoA, which is a 4-carbon compound, get converted into the succinate, and the enzyme responsible for this is succinyl CoA synthase or succinate thiokinase. Now succinyl CoA is a high energy compound. It contains a high energy thioester bond in its structure, and when that thioester bond is broken down, lots of energy is generated, and that generated energy is harnessed for the formation of the GTP from the GDP, and this GTP is then converted into the ATP and coenzyme A has released. So in this way succinyl CoA is converted into the succinate, which is a 4-carbon compound. Now student, in the sixth step what happens? Succinate is converted into the fumarate by the enzyme succinate dehydrogenase. Here again oxidation of succinate takes place, means the removal of proton takes place, but now student, here instead of NAD plus, the proton acceptor is FAD, flavin adenine dinucleotide, and hence FAD get reduced to the FADH2. Now in the next step, in the seventh step, fumarate get converted into the malate by the enzyme fumarase, and here hydration of fumarate takes place. Hydration means the addition of water molecule takes place to the fumarate and malate is formed, and the enzyme responsible for this conversion is the fumarase. Now in the last step of the Krebs cycle, malate is converted into the oxaloacetate, a 4-carbon compound, and the enzyme responsible for this conversion is the malate dehydrogenase. Here again oxidation of malate takes place, removal of protons takes place. Here proton acceptor is NAD plus, and NAD plus get reduced to NADH, and again oxaloacetate is generated and the cycle again runs. So now out of these 8 steps of this Krebs cycle, all other steps are reversible except the 3 steps. The first step is the irreversible step, it operates only in this direction. The third step is irreversible, and the fourth step is also irreversible. All the other steps of the Krebs cycle are the reversible. Now what we get at the end of the Krebs cycle? We get 1 ATP is generated in the Krebs cycle at the step 5. 1 ATP is generated. Then how many NADH are generated total in the Krebs cycle? 3 NADH are generated. 1 from the step 3, 1 from the step 4 and 1 from the step 8. 3 NADH plus H plus are generated. Then FADH generated. 1 FADH2 is generated from Kreb cycle at the step 6. And the CO2 released. How many carbon dioxide are released in this Kreb cycle? 2 carbon dioxide are released at the step 3 and at the step 4. Now remember 1-acetyl-CoA. For 1-acetyl-CoA molecule, this cycle runs only one time. So for 1 molecule of glucose, as from 1 molecule of glucose, 2 pyruvate are formed. And from 2 molecules of pyruvate, 2 acetyl-CoA are formed. So for 2-acetyl-CoA, this Kreb cycle runs twice. This Kreb cycle will operate twice for the 1 molecule of the glucose. So therefore this all amount has doubled for the 1 molecule of glucose. Means 2 ATP are generated, 6 NADH plus H plus are generated, then 2 FADH2 are generated, and 4 CO2 molecules will be released for the 1 molecule of the glucose. Student, I hope you understand this video. In the next video, I am going to explain about the ETC, that is the electron transport chain. Thank you so much. Please like and subscribe my video.